Hello everyone. My topic for discussion today is genomic level of regulation, histone modification, acetylation and methylation. So, what is gene regulation? Gene regulation refers to a set of mechanisms and processes by which expression of gene is controlled or modulated so as to get useful products. Gene regulation is critical for proper functioning, development and adaptability of cells. Gene regulation takes place in both prokaryotes and eukaryotes. However, this video focuses majorly on eukaryotic cell system. Gene regulation occurs at multiple levels, starting from genomic level, transcription, post-transcription, translation, and post-translation level. Here our discussion is majorly on genomic level of regulation, specifically epigenetic modification. Epigenetic modification is addition of chemical groups directly to the DNA molecule or the histone molecules. The chemical groups that are added are acetyl group, methyl group, phosphoryl group, etc. So, what is a histone? Histone is a positively charged proteinaceous structure around which negatively charged DNA molecule is wrapped around. It appears to be like a beads on a string model. Histone is a proteinaceous structure. It has four major subunits: H2A, H2B, H3 and H4. These four subunits have N-terminal and C-terminal. The N-terminal is rich in basic amino acids such as lysine and arginine, and this makes them the site for modulation. The different types of modulation that occur in histone are acetylation, methylation, phosphorylation, and ubiquitination. But about this, why is histone modification essential? Yes, that is an interesting question. Because as DNA is wrapped around histone structure, it is tightly wrapped around. So, as to do various processes such as replication, transcription, translation, we need the DNA to be in relaxed form. So, in order to make the DNA relaxed form, we need to modify the histone around which the DNA is wrapped. Therefore, histone modification is essential for replication, transcription, these basic processes to occur. Coming back to the topic, our video majorly focuses on acetylation and methylation. What is acetylation? As the name suggests, it is the addition of acetyl group to histone protein, and it is associated with relaxed chromatin structure and active gene transcription. Whereas methylation includes addition of methyl groups to histone and can result in either activation or repression of gene expression. So, acetylation occurs in three major steps: enzyme activation, chromatic relaxation, and gene transcription. Acetylation requires an enzyme named as HAT, histone acetyl transferase enzyme. As we can see in this image, the N-terminal, which is rich in lysine amino acid residue, is basically positively charged. So when acetyl group is transferred to this N-terminal ka lysine amino acid, the positive charge is neutralized by the acetyl group, therefore allowing relaxation of the structure and allowing transcription to occur. This acetyl group can be removed from the lysine amino acid by histone acetyl deacetylase enzyme, thereby again condensing the chromosome and transcription won't occur. The second method of modulation is methylation. Here, the key or interesting factor is methylation can either lead to activation or repression. This depends on the residue of lysine on which methyl group is added. So, in this structure, we can see that four subunits of histone: H2A, H2B, H3, and H4. In the H3 subunit, we can see that lysine residue is at 4th position, 9th position, 27th position, and 36th position. This position which I just now listed out are important. So, when methyl group is added at 4th position and 36th position, it leads to activation. However, if methyl group is added at 9th position and 27th position, it leads to repression of the activity. So, this is a key point. We can memorize it like even numbers. So, 4 and 36 are even numbers, so these lead to activation, whereas 9 and 27 are odd numbers, so this leads to repression. Now, there are various techniques used to study these histone modification. These techniques include chip sequencing, mass spectrometry, and proteomic profiling, definitions for which are stated above. Also, there are disease associated if these modifications are not done properly. The two major diseases which I came across are cancer and cardiovascular diseases. In cancer, histone deacetylation and increased methylation are commonly observed, causing repression of tumor suppressor genes. As tumor suppressant genes are repressed, this allows multiplication of tumor cells, which is not fruitful. Second is cardiovascular diseases. Altered histone methylation pattern can impact the expression of genes involved in inflammation, endothelial dysfunction, and atherosclerosis, contributing to heart disease. From this, I would like to conclude my topic. For more detail, you can refer Leninger 6th edition and Watson 5th edition. Thank you.